doing in the lab, as well as meet all of you and have everyone meet the community as well. So thank you so much for coming to today's Open Lab. My name is Giovanni. I'll be the hostess with the mostest, as Vanessa said. Without further ado, I just wanted to kind of tell you what the plan for the rest of tonight's night is going to be. We've been having some food and drinks outside, but for the next of the time, we're going to be doing a small introduction on Genspace, and we'll jump into the presentations that include the high school students at their iGEM project, as well as some of the member staff will be doing some presentations, and closing the night will be the community project with doing some quick lightning presentations. So with that, we'll finish with some closing remarks and then continue showing you all the cool stuff that's in the lab, and then just kind of hanging out and enjoying each other's company. So before we start, we always like to have events like this, and we like to keep them free. So if you guys could spare a little bit to keep these free, please, there's a tip jar in the back, and we have a site to donate on our website if you like what we're doing and want us to keep doing this. So thank you so much. So for some of you that may be like the first time here at Genspace, Genspace is the world's first community biology lab where we're trying to expose people to science in all the different ways that it looks. And we want to, more importantly, have this kind of rooted in the neighborhoods, just giving it access to everyone who's interested. Running the ship is our newest staff is creative programs coordinators. So this is Carolyn Hall. Is Clarinda here?
Yeah, she's here in the back.
Clarinda's in the back. Our development manager is Vanessa Fleury. I'm in the back as well. Layla Hernandez is our education manager. Layla. I'm the lab manager. And then making everything run smoothly and keeping us on straight and narrow is our great executive director, Elizabeth Tuck. This is Genspace. So we always get asked, how did Genspace come to be? And this is our origin story for our nonprofit, is that it kind of reads like a joke, right? An artist, a journalist, and a scientist all met in the Brooklyn living room. But they were all looking for a place where they can do science that was out of academia, that was out of big industry research companies, and they wanted to have something that could be shared and can be in the community. So they decided if the space is not there, why don't we make it? So 13 years later, we're still around, and we're still keeping to the missions and the vision and the mission, where our vision is to kind of have everyone have access to science, but the mission being always to kind of make a safe and inclusive environment where everyone can kind of learn, create, and grow with the sciences. So with that said, the way we kind of try to reach out the sciences in all those various forms is through membership. So for membership, you can have access to the lab to explore any kind of project that you're interested in. We also have classes and workshops where you can learn anything from coding to making microbial dyes and everything in the middle. We also have youth programming. So if you know a high school student that is interested in science, we have a program where we can take them for the summer, teach them molecular biology, and have them pursue their own project in our molecular biology lab here. And we also have outreach events. So you might see us at the Sunset Park Street Festival, where we're showing people how to extract DNA. We've flown kites with petri dishes trying to trap microbes from the sky, and then just seeing what grows. So we also outreach that way. So all of these kind of programs is what we kind of use to kind of get into the community and get science into people's hands. With that said, shameless plug, I am the lab member. I am in charge of membership. So if you're interested in learning with community, or have a project that you want to pursue, or maybe you're at that startup phase where you want this place where you can just learn and tinker and just be out of academia, this would be a great place. So if you're interested in membership, please find me, and I will gladly talk to you about anything involving membership, what we offer, and show you some of the things that we have here in the lab. So without further ado, we have the first group presenting is our high school iGEM team, and they're going to tell you all about their project. Oh. Okay. As they get ready.
That's good to hear. Okay.
So
Hi everyone. This is our project for our iGEM team. What is iGEM? You'll find out pretty soon. Our team name is PCBusters, and we're here to tell you just like a little bit about our project and what we're doing. So
our team name PCBusters comes from our target of the project, which are PCB.
Hold on, I think he's here. No, Max.
It's okay. He'll be here in 2 minutes. It's okay. All right. So our target of the project is PCBs. So what are PCBs? PCBs, also known as polychlorinated biphenyl groups, were first manufactured in 1930 and ended production in 1979. They had very advantageous properties for manufacturers and were very, very heavily used. But unfortunately, they were first detected in the environment in 1966, which was a major problem because of their hazardous nature. If you look at the molecular structure, you'll see that the number of chlorines will vary from PCB to PCB, meaning that they're not all the same, which makes it even worse.
You might be asking, why are PCBs dangerous? The dangers of PCBs originate from three main concerns: the stability, lipophilicity, and transmissibility of PCBs. PCBs are hazardous with their stability because they're unable to react and form theoretically more stable molecules. And their lipophilicity is hazardous because they range in consistencies from oils to waxes. And they are very transmissible because they're heavily breathed and ingested. In living systems, they are carcinogenic and can cause neurodegenerative diseases in women specifically.
So one of the major problems with detecting PCBs is that there isn't actually a simple and straightforward method to detecting them on the market right now. So our team decided to make a new plasmid that would act as a biosensor for PCBs through a luciferase reporter. The luciferase enzymes come from fireflies and emit light whenever they're in contact with a PCB.
So here's a simple quote-unquote diagram of what we have going on here. Just very quickly, so you'll recognize your good friends PCBs in the cell. So they are taken by AHR and ARNT into the nucleus. Does it have a laser pointer?
I looked for it, I couldn't find it.
Okay. Well, they're taken by AHR and ARNT, the green and dark green respectively, and bound to our circular thing. You can see with the star. That's a plasmid. Which, once they are bound, they start producing the luciferase that James talked about, and the cell glows in contact with PCBs.
Okay. Hi there.
Right inside.
So anyways, back to what Luke was saying. So essentially, an important thing for you to understand is that the AHR, ARNT complex, and they're pretty long acronyms, so you can see what they stand for on the top 2 bullets. They come from humans. So what we've been able to do is actually get their genes, and we've been able to order a plasmid that will express these 2 complexes. What they do, again, to just reiterate what Luke was explaining with those diagrams, is that in the presence of a dioxin, or a dioxin like PCBs, because PCBs are so close in like their chemical structure that essentially like the AHR ARNT complex will respond in the presence of PCB. It will take up the PCB, essentially binding to it. It'll translocate it through into the nucleus of a cell. And in the nucleus, it will bind to a special region of the DNA in the nucleus called a DRE, which is known as a dioxin responsive element, which we will explain more in further slides. But the point is that downstream of this dioxin responsive element, we have put in something called ACALU, which is the gene for luciferase, and luciferase, as James told you, is light. So that is how we quantify whether or not we can detect PCBs and whether or not they're there by essentially creating light.
Right. So just to quickly recap, dioxin responsive element. Dioxins are a similar class of chemical. They're quite related to PCBs. So the AHR and ARNT that we discussed about before will work fine for them. They bind upstream of our luciferase gene called the CYP1A1 gene, which is also found in humans. And what this allows us to do is basically say when PCBs are bound to this DNA, anything downstream of it will start producing its DNA. Originally, this was made to start degrading PCBs or dioxins within cells to keep the cell healthy and alive. But instead, we've hijacked that process to allow it to produce light instead, giving us a visual indicator of PCBs whenever the cell is in contact with them.
This all comes together in our PLK001 plasmid, named after the lunch. So it contains 3 components, which are digested PBF303 plasmid, which acts as our PCB biosensor and is pretty much backbone. And then the CYP1A1 gene fragments, which are the responsive elements, the human CYP1A promoter region, and the ACA loop, which is the luciferase, so that will create our light. And right now we're working on transforming this into yeast and having our PCB, having it act as a light reporter when PCBs are present. So that's what we've done. Literally 2 days ago. Yeah, ready to talk about that.
So yeah, here's this is our PCBusters team that we set up. Okay, so when we came up with this project, we had very high expectations, and something we found very quickly is science is slow, and deadlines come a lot faster than you think. Versus like next week. So but we still like this project like a lot, and we invest a lot of time into it. So further things we would like to do in the future first are to find out whether or not the plasmid that we make actually kills the yeast that is put inside, and just make sure that the yeast are happy and healthy and can survive with the plasmid in it.
Yeah, the next thing that we'd like to do is grow our plasmid PIGHARC, which is an acronym for something really long, in galactose, which would induce the expression of AHR and ARNT.
And we're also hoping to eventually test our dual plasmid system in the presence of a PCB or a dioxin on a plate reader. And the plate reader would tell us whether or not the plasmid actually worked.
And then finally, the last thing that we are really excited about is the idea of creating something called a luminometer, which would work in place of the plate reader, and we could do this with a circuit board, for example, that could quantify light, and ideally we'd be able to just create a circuit board on a hardware section of this project, and that way we can make it commercially feasible to use and can make it accessible to everyone in the world to be able to quantify whether their water sources are contaminated or not.
All right, so yeah, that's our project.
Also, thank you to Beth and our mentors,
Yes,
Casey and Debbie.
absolutely. Thank you.
And our sponsors for collaborating with us.
Did you guys go to the Jamboree yet?
It's next week.
It's next week. Yeah. it's
There's
next week.
time for some questions if anyone has
yeah.
questions.
Oh yeah.
Oh,
Go ahead, questions?
At the high school level, do you have to submit biobricks as well?
Yeah, we did.
Yes,
We did.
They're not easy to do.
They are, yes. So BioBricks, for anyone who doesn't know, BioBricks is iGEMs, I believe,
Registry?
yeah, registry of compatible DNA. That this was sort of a system that was set up back when DNA hadn't progressed as far, i.e., 5 years ago, which really shows you like how far this stuff progresses. So at that time, it was a lot harder to piece things together. So you ideally wanted DNA that you could kind of snap together like Lego bricks, like BioBricks is in the name. However, since then, we can manipulate the DNA so much easier that becomes a lot more of a hassle than is really a help now. So yeah, that is, but we still have to do it because we have to be good scientists.
Contributors. The iGEM
Yes,
world.
yeah. Yep.
Why'd you use luciferase and fluorescent protein?
Oh, sorry, I can.
Go ahead.
Go.
Yeah.
Well, in our research, the only other biosensors that exist use GFP, green fluorescent protein, and we just thought that because this was the only biosensor that exists, if we want to create new ones and just add to just the general amount of data just on biosensing, we wanted to see if a luciferase assay would be more efficient and just see what we could do with that just to add to the amount of data people have on these types of things.
It's more sensitive, too.
Mhm.
Yeah.
You have a lower threshold of expression.
Awesome.
Loafers in the back.
Yeah, I guess I'm just curious, you said there aren't a lot of other sensors for PCBs
No.
out there.
There aren't. So,
Is that why, is a biosensor, is that an ideal?
yeah. So right now, the only method is using chromatography. Right?
There's high resolution gas chromatography and then high resolution mass spectrometry, I would say, and then there's some diamond gold nanocomposite after sensor, which are big words for very expensive things that only trained personnel can use, and the only big entity that is fighting against PCBs right now is the EPA, and the EPA only targets large corporations like General Motors that it has been known to dump a lot of PCBs into the Hudson River. We wanted to target just the average people who have contaminated water sources that don't know that, so we wanted to create something that could be used for them.
Yeah.
So,
Yeah.
so you're saying that you're going to commercialize this by attaching a circuit board?
Ideally.
Ideally, yes.
Using like an Arduino sensor.
Yeah.
Yes.
So something very cheap, something very affordable. The idea is, get it out to nonprofit groups, local community advocacy, all that fun stuff, and just have something economical with a sensor that you can grow and a circuit board that costs nothing to make, and it's just very easy to set up, and basically sidestep the whole $10 million machine that only exists in university idea.
Sounds
Yeah.
great.
Yeah.
It sounds great.
Yeah.
I'm just curious, how long do the sensors last? Because they're living systems.
Well,
We haven't
So-
gotten to that.
Yes.
I want to get to that. Is if the yeast gets transformed, we kind of did that 2 days ago. But if it works, our next goal would be to do a toxicity assay,
Yes.
which would be, does the yeast even survive with it there? And then if it does, we can test how long it survives.
Okay.
Yeah.
Excellent.
Yeah.
Sounds good. Thank you.
Yeah, can we give another hand to the Genspace? Thank you so much. Next up is a member, Sam and Emma from Envirobe. So, Sam, take it away.
Thank you. Where's the laser pointer? Is this guy? It's like the button. Yeah. Good evening, everyone. So my name's Sam Magaziner. In my day life, I'm an MD PhD student at the NYU School of Medicine, but at night I moonlight as an entrepreneur for a biotech company. And I suppose since this is about Genspace, Emma and myself have been members in the premium category for startups for just over 2 years now, 2 years and 2 months. So a long time. We actually started during the pandemic, and there was only us and then another premium membership. So it's a very different space than it is today, where there's a lot of collaboration, a lot of community projects ongoing. And so the company that we work on here is called Envirobe, which is a portmanteau of environmental microbes. And so our venture is based around biochemical and biocatalytic means of PET plastic breakdown. So I apologize, this is a bit busy, it's normally animated, but let me walk you through it very briefly. Most plastics begin their life as fossil fuels. These fossil fuels are actually turned into chemical precursor monomers, single chemical elements, that are then turned into plastics that you know and use, like single-use water bottles and single-use packaging for things like vegetables. These PET products then find their way to you, the consumer, and the consumer has a choice. The consumer can choose to send it to a landfill, which inevitably will end up in the environment over time, or they can be sent to a recycling center. At these recycling centers, the plastics are sorted and then sold as commodities to processors. In this case, we focus on PET plastic, which is the most common type of consumer plastic. PET processors will then take this plastic, process it, grind it up in a very physical maceration, melt it back down, and then shuttle it back to PET manufacturers. The issue, however, is twofold. First of all, PET processors themselves generate much byproduct, which will also find its way back into the landfill in the environment. Likewise, this process is not infinite. Plastic that is mechanically recycled can only be processed in this way about 3 to 4 times before it really loses rigidity, clarity, and general purpose use. And so Envirobe set out with the mission of finding a way to infinitely recycle plastics in a biological-based manner. So many of you might be familiar with this study. This came out in 2016. A group of Japanese scientists isolated a bacteria that was capable of degrading plastic called Ideonella sacchaensis. I know you've used it in the lab. But the issue with this bacteria and why it's a poor model for actually being a biological means of breakdown is very little is known about it. It's limited studies, it is unknown pathogenicity, it's incredibly slow growing, and its inherent breakdown rate is very low. In addition, there are no genetic tools. Some other companies and some other scientists, since, and mostly at universities, have sought to use an enzyme-only approach, much like how you might make cheese, where you take an enzyme and you add it to a batch, and then through biocatalysis, the cheese is formed, or in this case, the plastic broken down. However, when you're dealing with recycled plastics, this is incredibly rate limiting at an industrial scale. Recycling plants deal with upwards of 25,000 tons of plastic annually, which on the scale of biodegradation with an enzyme would equal thousands and thousands of dollars. So put in other terms, an enzyme-only approach where you take this enzyme that breaks down plastic and put it into plastic itself is not scalable and not efficient. In essence, it's not cost-effective. And so what Envirobe has spent the last really 3 years doing now is generating an efficient microbe. A microbe that's capable of reproducing at a high rate, that is non-pathogenic and safe for industrial use, and that is self-sustaining. This means that if you take plastic and you put the microbe in, it'll grow on its own without the need for additional enzyme additives, without the need for glucose or other carbon supplementations or other amino acid supplementations. And likewise, we hope to integrate this seamlessly into a processor environment. That is done by this bioreactor, which is a large-scale mock-up of a small-scale mock-up that's located in my friend's house in Staten Island. Luckily, I haven't had to move it here yet. But Envirobe's practice is twofold. We've been generating this microbe through both directed evolution and metabolic engineering for the utilization of these plastic carbon sources, and the utilization of this bioreactor. What this allows us to do is an infinite upcycling of plastic, whereby the bacteria can turn the plastic back into its chemical precursors. These chemical precursors can be shuttled back into textiles or be used to create virgin plastics, that is, plastics which have the same properties as brand new material. And what's particularly interesting to us is that because we're working on a biocatalysis level, we don't care about the mechanical filtering and sorting that traditional recycling does. What this means is we're able to capture plastic feedstocks which are currently inaccessible to modern-day recycling technologies, including and not limited to ocean plastics, which in recent days has become a widespread problem globally. And this allows for an infinite process of upcycling because every time we convert it back into chemical precursors without going through that mechanical recycling current processors do. I think that's everything, yes. So, questions?
That's amazing, first of all.
Thank you.
Thank you. I was wondering, so in your comparison to the mechanical process, how much time does it save you versus when you're like breaking it down basically by solvents in the mechanical system? Like, what's the reduction on that time scale that makes this more viable?
So it's not just time scale. In fact, the time scale tends to be around the same. But what we save on is carbon emissions, because the process of mechanical breakdown requires large heat input. They wash the plastic through high temperature baths, and they have to heat up this plastic to incredibly high temperatures to melt it back down into the pellet resin form that gets distributed. Likewise, we can recapture these unutilized feedstocks of plastic. And so that's the primary value proposition, in addition to the process of a more cost-effective breakdown. So it's both environmental and cost-effective, which for anyone that's doing really startups that are focused on both the environment but also have a biology focus, when you talk to investors, if it doesn't make money, they won't care, which is the sad truth. So things have to be both environmentally conscious but also cost-effective and at the end of the day scalable.
And you do both.
Well, try to.
So I had a question. I'm not really familiar with all of this, but so before you put the enzyme or the microorganisms in contact with the plastic, like, do you need to do any like preparation, right? Because I'm thinking about like surface area. Like, how does that look like?
It's a good question. So what's unique about our system is the bacteria are never in direct contact with the plastic. So this avoids issues of contamination, right? Because you can imagine that recycled plastics are incredibly dirty with food residues and God knows what else. And so the way we've designed our bioreactor system is that we have a plastic feedstock chamber, which is being constantly fed new plastic feedstocks that is then attached to a bacterial chamber. The bacterial chamber secretes effluent that is then introduced to the plastics. And so by separating the biological process from the degradation process, there isn't really any complicated preparations needed, outside of perhaps washing it and keeping the temperature of the water around 30 degrees Celsius, just a more efficient temperature for the enzyme to work at.
So you need to like chop it or pulverize it?
No, so more surface area inherently helps. And it may come one day when we scale to a more industrial pilot that needs to happen, but not at this time. Yeah, it's a good question.
So a lot of plastics have preservatives in them. So when you have the feedstock, do you have any way of controlling? So, or actually, the quicker question is, does this organism have any resistance to like preservatives currently used out there in plastics?
It's an interesting question. So no, but we haven't run into an issue with it yet.
So there's no like diminishing of the population, the bacterial population.
So the beautiful thing is if the bacterial population crashes, I just go into the freezer, grab a little tube of bacteria and dump a new one in. That's sort of the self-renewal element that overcomes issues with an enzyme-only degradation system. Yeah.
Yeah, thank you so much. I will encourage you to find Sam and ask him more questions. He'll be around, right?
We'll be around. Yeah.
Yeah. So another hand for Sam is Envirobe. Next up is two of our very own. Carolyn will be telling us a little bit about her project.
So who wants to time me? I'm the timekeeper. Who wants to time 5 minutes? You literally, great. Yell out 1 minute at me when we have 1 minute. All right? All right, here, I'll just give you my phone.
Yeah, it's fine.
I love it. Okay, audience participation already. Hi, I'm Carolyn Hall. Clorinda MacLowe is in the back. We, in the red sweater. We come as a package, what we do. We are the, as Giovanni said, we are the new creative program coordinators. But we also do a project called Sunk Shore, which is about connecting people to climate change on shorelines. And this all started, this all started because of this question. Why is it related to climate science and climate data, really? Like there's so much data out there. Why is it so hard? Why is it so difficult? Why do people have such a hard time doing it? Well, one of the reasons why is because people are shown things like this. Now, people in this room, probably most of us are science literate enough to understand at least the gist of this graph. But you have to be able to understand what axes are, what the variation is, and to see all this variation, to see a general trend, right? Like, there's a lot to understand here. And if you do not have a science background, this is completely overwhelming by itself, let alone when you get this, right? This is what we get when we hear the media talk about climate change. It's dying polar bears, it's floods and fires and drought. How is it flooding and drought? It's all these different tables and all these different charts. It's projecting into the future, it's looking in the past. It's a lot. The data is a lot. So, but we need to understand it. So, the next question was, how can we help people make this climate data become personal? How can they feel it? How can they relate to it? How can they understand it so that they would want to do something about it? So they would see it in their own homes. Well, the first thing with Sunk Shore, which is an experiential time-traveling walk along shorelines, is you make it local. So we take people to their shorelines. We've done this on a number of shorelines in New York City. And we do a tour that works with the senses so that they understand the history. They know that people know the history of their own shoreline. But they also can feel it in their senses. Like, what did it feel like or smell like or sound like in the past? What will it feel like, smell like, or sound like in the future? Right there, where they're standing. So you make it local to what they already know, what they're already experts on, their home. You make it active. So here is myself and Clorinda with our tour group, and we are in boats. So the reason why is because the data in the future is that there will be so much storm surge and so much sea level rise that this
sidewalk will be underwater. So you can't walk in the future, you can't walk on water. Well, we haven't figured that out yet. But you can row a boat. So we have props that we make easy to handle. We make people be cooperative together. This was pre-pandemic. We have fans which become oars, which become sunshades, which become fins. And we make people do things so that it actually becomes part of their physicality. The reality of a future becomes part of their physicality. The data of storm surge and sea level rise becomes part of their physical experience. So we make it active. Engage the imagination. Here, people are holding a card in front of their face. Why would you do that? You could only use your peripheral vision. What creatures who live in water see out of the sides of their heads? Fish. So maybe you have to learn how to navigate like a fish. Okay? Like, this is the thing. Use your imagination for playful ways like this to think about a more aquatic environment, because in New York it's going to be a lot more water. Or you can look at a building. I have 1 minute. You can look at the building and see that it's flooding. Thank you. And in the future, if a building is flooding all the time, what can it be instead of, I don't know, a school? It could be an aquaponics center where you grow food for the community based on water. Okay? Use the imagination. And finally, make it fun. Here people are navigating a corridor. Actually, it's a time portal, but you wouldn't know that in this picture. From 1 year to the next year as fish, which was required because it was a water portal. So we do this also. We teach these ideas, this idea of like, how do you make the future that's climate crisis, for lack of a better word, how do you make it, how do you teach how to think of it in a creative way, and in a way that can actually be applicable and interesting and not overwhelming and matter to you and make you want to innovate a future that's better. We teach workshops. This is sort of the imaginative moment and where people are starting to like connect to water lines that matter to them. And this is taking people out. This was a Genspace workshop, by the way, earlier this year. And this is I'm done, I see that. This is taking to shorelines and have them give a tour. They last. We also do installations. This is sea level rise, 2,100, 6 feet higher. What does it mean to stand underneath it? And finally, we do other kinds of art. As many access points as possible. Maybe there will be flamingos in New York. That's it. Thank you, David.
Any questions for our speaker?
Great.
I have a question. What inspired you to specifically focus on the shoreline?
So I am a marine ecologist also, and my work is in fisheries, and the history of fish in New York City has been a big study of mine. But also is historical ecology. What's the use of a shoreline in the past? How has that changed the ecological nature of that shoreline? Basically, what humans have done to shorelines, how has that changed the balance or unbalanced the nature? And where is that going in the future? So that is my personal history, and Clorinda MacLo, also in the back, is very much about ecology and place. And we care a lot about, and she's a New York native, so she's watched these shorelines change and change and change. So we care a lot about shorelines. Any other questions?
Thank you.
Thank you.
Next up is
Yeah, and I have these to pass out if you want. These are scores for if you were to go on this and you want to have an experience. Highly recommend. This is so cool. I have goosebumps.
yeah.
We're Jess. Hello. I'm Jess Thies. I'm doing my MFA at Parsons right now in industrial design. And this is a project I was working on last year. So I guess I'm presenting today my work with Chlorella vulgaris. And this is a microalgae. It's a single-celled organism. And I was really interested in this concept of carbon capture. You know, I think it's something that people are talking about, and I think I was looking at, well, what are the ways we can do that biologically? And so I kind of got a culture. I was growing it in my apartment. It grows in like 20 to 30 degrees Celsius, 6 to 9 pH. It's pretty adaptable. So, and these are some like pictures of it under the microscope. And so I think starting off looking at like its carbon capture abilities, I started looking at other designer projects. I'm a designer. I wanted to know what other people were thinking about it, and the possibilities of it being a protein source for food in the future, and also people thinking about it as biofuel in the future. And how can we kind of work and collaborate with this organism to kind of create a different future? So I kind of started doing research more into microalgae, and chlorella can absorb carbon up to 200 times more than trees can. They photosynthesize, so they absorb CO2 and then release oxygen. And this one particularly has a dry oil content of over 50%. So it's a really great microalgae to use for biofuel production. And so these were kind of the things I started looking at. Like, could this be a possibility to help us curb our carbon, or the way that we have carbon dioxide in the atmosphere? How do we collaborate with this living system? And so I designed Envirobe. And so this design is an at-home carbon capture system. So the premise is that it would actually be, self-sustained in your home. People could do this themselves. So there's like 3 objects in the system. So there's a bioreactor where you would grow your microalgae. There's an oil press where you would process the harvested microalgae into an oil, and then and this an oil lamp. This is like kind of a proof of concept that you could burn the algae and kind of create it into a fuel that way. So it's creating light, for instance. This is just a kind of rough prototype of it. But essentially you would place your algae culture, any nutrients it would need, like phosphorus and nitrogen typically, into the bioreactor. And this design is kind of based on like a bubble column reactor. Not working, but and after growth, then the algae can kind of be filtered out and harvested and dried out. And then it's kind of placed within the oil press, where it's heated up and spun through a screw press, which extracts the oil from the extracted algae. And this is kind of similar to like how olive oil is made. Like I was kind of looking at what are other ways oils are produced. And then that could be placed within your lamp and like lit. And who knows what else it could be scaled up to be. And I did a lot of research, like talking to microbiologists, some of which were part of Genspace, so thank you for that. And I think that I really was looking at, what are the forms that these different experiments could take? How do these fit into the home? How do they, how do they aesthetically fit into our homes we have today, or what do our future homes look like? Those are kind of things that I think are interesting to explore in the future. And also, this would need a lot more testing to really. I would love to do a prototype with real materials at some point to further calculate like how much algae is needed to grow for production. I don't really have those answers yet. And really specific like timelines, like how long would you need to grow before you're harvesting it. But they're all interesting things to explore, I think. And yeah, I think the purpose of the project was really to reanalyze like how could these biofuels like live within our homes, and decouple us from the petroleum-based fuels that are, run by bigger companies. Could you like have this self-sustaining system yourself? Yeah, and I think that one issue is that, well, if the point of the project was to carbon capture, but then you're burning fuel, it kind of doesn't work that way. So that's the end. But I'm still kind of playing with algae and working with algae for future projects. So we'll see where it goes.
Yeah.
Thank
Any questions for Jess?
You mentioned the fuel applications. on a scale of like a candle to a car, where do you see them being applicable?
Yeah, I think that for you to do it in your home, I don't really see you being able to do it for a car right now. But who knows? Like, I think that we would really just have to test it and see, like, well, how much are you producing oil content versus like how amount of algae you're growing and how fast is that growing?
How's it smell?
Ooh. Does it smell? It doesn't smell that much. I actually have some here if you want to see it. But yeah, it doesn't really smell. It's fine.
Have you tried mixing different smells with the algae oil?
Ooh, I haven't. No, but that would be fun to play around with. Yeah.
I'm just curious, like, you have to like agitate it, like keep it alive.
Yeah, so I think that you would have, the way that I was doing it was like I had an air pump kind of like, circulating.
Yeah. Do you ever get contamination from things that are photosynthetic that might make it smell?
I haven't had any contamination, I don't think. I mean, we've like looked at it under the microscope, so maybe there's other things in there, but it's still like green and growing like after a year, so it's pretty resilient, this particular algae.
You've been focusing on use as an energy as a food source?
I have, yeah, mostly. I think I'm actually kind of transitioning a little bit to future implications of keeping it alive. Like, I think that's something I'm really interested in, like photosynthetic, the photosynthetic properties. Yeah.
Have you eaten it?
I was like, I have like the dried spirulina, I've eaten that, but people are like doing that for protein shakes and stuff. I don't know.
So can we get another hand to Jess? Next up, we'll be having a few community projects give some lightning talks. So up first is the Open Plant Community Project. So look at that.
Hello, everybody. Wow, there's a lot of you here. That's great. So I'm going to talk a little bit about Open Plant. Tina, our project leader, is not here. Unfortunately, she's recovering from COVID, so I'm filling in for her. So the project is inspired by the research initiative out of Cambridge in Britain to make oh, I'm sorry. No, wrong button. There we go. Sorry. To develop the common liverwort plant, the Latin name is Marchantia polymorpha, into a model organism, and to develop it for synthetic biology. So, inspired by that, we would like to express insulin in Marchantia. Why insulin? The insulin market is currently monopolized by 3 large pharmaceutical companies, Eli Lilly, and I forget the other 2. And the price doubled between 2012 and 2016. A lot of people in the United States are actually self-rationing their insulin, not getting enough insulin. And there's too slow, I got to speed. Okay, so let's go on to the next slide. Yeah, so that's the reason I want to do this. What have we achieved so far? Well, from right to left, building a DIY incubator, which is right over there on the shelf there. During the pandemic, when the lab was closed for $200, it's got a bunch of nice off-the-shelf parts, Raspberry Pi camera, LED light strip, temperature humidity sensor, cooling unit, etc. And there it is. I call it piano bench because it was on my piano bench. We've done molecular cloning and transformation of Marchantia with the Ruby reporter system. You can see right here. a purple plant and a close-up of a purple plant. It's purple because it has been transformed with the gene that comes from beets that is responsible for their purple color. 1 minute. Okay. Wow, this is fast. Oh, I just want to say, why do you need a reporter system? You need a reporter system so you can easily see that the plant or the organism is doing what you want it to do. Otherwise, how do you know? Okay, what's next? Yeah, I think if I could actually look at this, right? So, we're ready. Move on to insulin. We're going to do molecular cloning, put the insulin into our construct, into our vector, transform plants. What else do we need? We only have that one little incubator there. We need bigger incubators. So, David, where is he? Got some big styrofoam boxes from the fish store. We're going to build bigger incubators. And develop a light box to photograph the plants and be able to automatically measure the growth, take a picture of it, upload to the cloud, computer vision software to measure the size, and then be able to plot growth curves. And yeah, come join us for science and fun with plant biology. I can't see a polymorpha growing in that incubator right there that we took last week. Any questions? Oh, the plate. And Dave has a ruby transformed liverwort right there. It's dried out, but you can, if you look closely, you pass it around, Dave, you can see that it's purple and not green. So, questions? Oh, go ahead.
Yeah, so what organism would you be getting the insulin gene from? And has anyone ever done insulin in a plant before?
Yes, they have done insulin in tobacco and I think lettuce leaves. I think it was done as a transient transformation. They did not transform the germ line. What was the first part of your question?
It's just where you get the insulin gene from.
Oh, it's, that's a good question. I don't know where ultimately it came from. I think it's probably synthetic, but we got it from the Open Insulin Project, from somebody in the Open Insulin Project. So we actually have it in the freezer here. It's very short. It's like 60 amino acids.
It's human, right, Jerry?
It's actually it's glargine, which is.
But it's basically human.
It's human insulin.
Yeah.
Yes, that's right. Yeah. Anything else? Oh, go ahead in the back.
Have you thought about looking at viruses and bacteria that actually do horizontal gene transfer into plants as a means of infecting plants and putting the human insulin gene into plant systems and not having to develop a model system overall?
I don't think we thought that. I mean, what we use for the transformation is Agrobacterium, Agrobacterium tumefaciens, which does the horizontal gene transfer, but we haven't looked at viruses at all. But that's interesting. Maybe you can talk to me more about that and understand more how that works. Go ahead.
Would having this plant grow insulin impact its growth speed at all?
That's a good question. We want to study that. Yeah, so we would compare with wild type and see potentially it could. Yeah. I think we noticed with the Ruby reporter that there's some impact on the rate of growth. When we get this working, we'll I didn't put this on the slide, but we would want to delete the reporter gene because it's just going to add metabolic load to the plant that we don't really want to have there once it's all working.
Oh, so you are going to modify the same plant that has the reporter gene. You're not going to modify a separate plant?
No, we're starting again from cloning and infecting another plant, and so we'll have both the reporter gene and insulin. But then in the future, we would delete the reporter gene, knowing that it's actually working. Yes, sir.
Question: when do you infect the plants? When they're not the seedlings, or bigger?
Yeah, very small. There's two methods of transformation. The Marcantia can produce asexually, and they produce gemma cups full of gemmi, which are clones of the parent plant. You can infect them at that stage, or you can get spores, which are the result of sexual reproduction. And at the sporling stage, when they're very small, you co-culture them with the bacteria infect them and do horizontal gene transfer.
Thank you very much. Highly encourage you to find Jeremy and ask him more about some of the fun. Up next, we have the Biomaterials Studio.
All right, biomaterials.
How does it work? That one's okay.
Yeah.
Feel free to look at that, and I guess, yeah, this way. Zigzag it, fill it out. So, let's see. Testing out. How does the laser pointer work? Oh, that one. See. Wonderful. Okay, that's great. Hello. My name is Nico Rance, and I'm the current lead for the Biomaterials Studio at Genspace. Thank you. So the formal brief that we have, the Biomaterials Studio is a community-driven biomaterial research group at Genspace where we focus on biomaterials. We want to explore, create, and innovate the intersection between materials, biology, and design. We play around, we explore, we push the limits to biomaterials, and we share that knowledge with all the members or anyone who wants to visit. For anyone who's not aware of what a biomaterial is, it's a material where some pre-existing biological component, i.e. some existing piece of wood or other like natural quote-unquote material, undergoes processing to become a useful fiber, fabric, or biocomposite. And this could be anything. The potential is limitless. The historic material that we're mostly all familiar is with wood, and people are still creating new materials with it. There's actually a company recently that was able to make wood into a clear, kind of like plastic material that could possibly replace packaging. There is a whole process with infrastructure and actually going through the systems of that. But regardless, there's other materials too, like fungi. You can see down here, right with the laser pointer, fancy. Fungi-based material. There are seashells, or materials similar to seashells that people are recreating. Gelatin, microbes, algae. Again, the possibilities are endless. At the Biomaterials Studio, we've been focusing on a few things recently. Gelatin is something we've been really fond of because it's really easy to work with these proteins. Specifically beef gelatin. We can either create a bioplastic you see on the left or more of a foam to the right. The skeleton head that you're looking for, or looking at currently, was inspired for Halloween prompts, and that's based from beef gelatin. We can actually rework that into a foam or into a flexible bioplastic. We've been having fun with that, as you can probably tell. Other things we've been working with recently is cornstarch, where we discovered that actually it doesn't lose its flexibility. It's still very flexible, or even liquid, you could even say, after weeks. And we've been experimenting, mixing that with other materials. Mycelium, which is based off fungi. One thing, let's see if this will work. A recent one is an iridescent patterning. Hopefully this will load, but you can also see the real sample in the lab after all of these, interviews, but all these like patients basically, and see them in person, but it has like an iridescent tint. So long-term goals, we do want to create a materials library that other people can have access to, in addition to current existing ones, but ones that we've been experimenting with. Finished products. A recent project, more short-term, is actually creating a light fixture for that light bulb over there. Briefly, it's very bright. You don't have to look too long. You can look away. We actually want to create like something that we've created and grown in this lab and actually put it right there as a showcase. Again, this is bio-related, biomaterial, so it might not be long-term, but that's okay. We can actually rotate it. Intentional. Exhibits online and offline. Long-term goals is actually having it someplace like maybe public art, maybe something else. We're open to it. And an open source book, again, back to the materials library and workshops. So if you'd like to be part of this, definitely join in on, we rotate between Monday and Wednesdays. Usually at the beginning of the month, it's a Wednesday, and then we shift up to Monday between 6:30 to 9:30. And that's a wrap. If there are any questions, please let me know.
if you have your own, we invite people with their own criteria to come over, and we can hopefully provide some information for your own project. For ourselves, it's seasonal, as you can see with the Halloween props and the biolab. It really depends on what we want to focus in the moment. We usually try and finish or create like an end product by the end of the material, even if there's other endless possibilities to go into. But again, open-ended to give you an answer. Any other questions?
What is your iridescent material made out of? Is it a bioplastic that has iridescence?
I can fill you in more about that afterward as well. If Giovanni, you might have an answer for that.
It's a micro patterning, so it's not a material property. It is a physical property. Yeah, there's fine structures on it that bend light itself. Yeah.
And we can, if you're interested, definitely hang around afterward, and we can tell you more about it. Any other questions? Shifting a little bit. No, at the moment. To wrap it up, there are more samples over there. We can give you the rundown of what we've created recently. Feel free to check it out. And that's a wrap. Thank you.
Thank you, Nico. Next up is like one of my pet projects for Genspace. It's the electronic upcycling. And this year, we've been looking for different parts, and it was actually the year of 3D printer parts, because in this space, there is a 3D company that has thrown away tons and tons of 3D printers, so that means we have tons and tons of parts. So once we saw these in the trash, we had to pick them up and see what was inside. So me and a member of Kamal started taking these apart and seeing what things we can pull out of it. And this is essentially what kind of drew our eye from these materials. We have power sources, we have these guide rails, we have these motors, and this gantry I'm not entirely sure what it is, but I knew I wanted it. So with all of these things, it was easy to start exploring what we could do with some of these materials that were just being tossed away in the trash. So from these things, I imagined we would make motorized microscopes. So all these motors are stepper motors that have fine control. We could easily add these to a microscope and add new abilities to our microscopes. The other setup that we saw here, this guide rail, I easily imagined it to be a syringe pump. And syringe pump is a gateway to multiple projects that include bioprinting, electrospinning. Essentially, you can make nanofibers if you give it enough juice, electric juice, and then microfluidics. So it'll give you fine precision of a small syringe. So with all these things, our goal is to use these materials to make some projects and add possible instruments that could help other projects. For example, we can do materials testing or tensile strengthening for some of the materials that the Biomaterials Club or BioStudio is making, as well as my pet project that I really, really want to see at some point is a 3D printer that hangs off the roof and it can print giant things. So with some of these parts, we can do that. But essentially where we are now is that we were able to scrap some of those motors and we were able to attach them to a microscope itself. And actually, thanks to Nico, who just presented, he was able to kind of do some CAD drawings and actually 3D print it. So we now currently have a motorized microscope here at the lab. Another thing that we were playing around with is that guide rail that we saw. We actually did make it into a syringe pump. So now we can control the precise amount that comes out. And then this kind of gives us access to microfluidics. So we can start doing smaller tier projects that can help us separate cells, can help us look at different types of algae. And the sky's the limit. And I've also been playing around with potential art we can do with it. So you can imagine putting chlorophylls in there and actually mimicking a leaf, changing colors in the fall within a few seconds. So thanks to Nico and his brilliant design, we were able to make the syringe pump, and now we have a suite of projects that I can't wait to get more involved into. So if you're interested in just upcycling and learning more about these electronics and just kind of imagining together, please find me, and I'll love to show you some of the things that are actually here in the lab. I'd love to show you those things as well. So thank you so much. Any questions? Yeah.
Just a fun thing to Google, you might want to look at gigabots. They don't hang from the ceiling, but they're pretty cool. And you got a micro center right here, so you can kind of just look at the little gantries that you got.
Yeah.
Go over there, buy some bigger parts, and then slap the servo motors on them. You can make a big 3D printer.
We have all the parts there. That's something that we want to do as well. I've also seen the one, that infinite 3D printer, the one that's on the thing. That's something that's on my my wish list as well. If anyone's interested, I would love some help with that. But yeah, that's on my list. Any other questions?
You made a motorized platform. Is that what that?
This?
Yeah.
So this is a motor that's attached to the body of the microscope.
Yeah.
It'll be changed to the fine adjustment. So
Take me a second.
yeah, so we just have to rotate it and it'll slowly do adjustments. So in theory, we can have like an image like a focus point here, rotate it, focus point here, and we'll make like 3D images of
Cool.
microscopes. The slides. Yeah.
It's like a manual these days.
Yeah.
Yep. Exactly it.
And we have fluorescence capabilities too, so now we can really run with this.
Cool.
Yeah. Very cool. Thank you so much. Up next, last but not least, is the Engineer Matters Lab.
Good job.
Go Jazz.
How do you?
The slides.
Oh, is it right?
Yeah. And
So we're Engineered Matter Labs, right here. Essentially, what we want to do is we want to democratize molecular biology so that way everyone has access to it, and in the process create sustainable materials for the future. Our first focus is spider silk. So why spiders and spider silk, besides it being spooky season right now? It's been the holy grail of materials for scientists for decades, and I mean, spiders are the original material scientists. They've been around for 400 million years, and they've been able to perfect this incredibly strong but elastic material that can hold almost 100 times its weight. So it's stronger than Kevlar, it's stronger than steel when you compare it on a weight-by-weight basis, so the implications are vast. The processing can be done at room temperature, which is pretty much unheard of when you talk about processing for materials in general. It is biocompatible and biodegradable. The applications are incredible. So textiles is a direct one that most people think of. So currently silk is made from farming silkworms. Ideally, the thought was to farm spiders, but spiders like to eat each other, which we found out with our 2 babies that were in the same area, only one was left. So they like to eat each other, so as you can imagine, you can't really make a ton of silk with one spider. So the idea was how can we create silks that are more sustainable and that are actually better than silkworm silks. Medical implications are crazy, so you can actually put spider silk within the body and neurons are able to grow along it. So it can actually regenerate those nerve endings essentially because it is biocompatible, and then essentially it'll just degrade over time. And then beauty. So potentially we're thinking of it as a replacement for collagen. If you don't already know and you're buying collagen, don't buy it. It doesn't do anything for the skin. It's just overpriced. You heard it here first. So that's another application. This is one of our babies, secreting spider that we've captured here in the lab, or secreting silk that we've captured here in the lab. We have 2 different avenues that we're currently tackling. The first is more basic science route. The second is a synbio route. What we realized was that even though over $1.5 billion has gone into synthetic spider silk companies, there's only 9 fully annotated and analyzed genomes out there for spiders. So there's a huge gap between people wanting to go directly into synthetic spider silks without actually understanding spiders. So one of our, hopefully, end of the year 2023 goals is sequencing and annotating a full non-orb weaving spider genome. So if you want to be a part of that with our new sequencer, please join us. We'd love to have you. And then the synbio route is essentially taking the info that we gained from the basic research route and being able to customize spider silk because it has these just properties where it's like little mini blocks that you can play around with that can make it more strong or make it more elastic. So from what we learned from understanding spiders in general, we can then better engineer synthetic material. And we do that through yeast, which is induced from the presence of methanol. So member experience, you get to do fun things like field work and catch spiders. With us, Joe is currently in Spain catching some peacock spiders. Hopefully we'll send us back some wolf spiders. We're just really cool people who do who like to do molecular biology.
We meet on Sundays, 11 to 1. We also meet throughout the week. The best way to keep up to date is go to eml.pubpub.org. We're going to update that every month. If you want to learn about spiders and the 45,000 species that exist, go there. And then if you want to contact me, you can email me. I would actually super recommend, so there was this huge paper that came out last week. They were able to actually capture over 1,000 spiders, and it's the most in-depth paper on spider silk that exists to date. I think it took 6 years to do. They did like over 8 or 9 countries in order to get the results for that paper. We're going to be talking about it with Joe from the Hayashi lab. Cheryl Hayashi is like the queen of spiders. She's been studying it for the 80s, and she said, the more you study it, the more you look like a spider. So I'm like, oh God, what's going to happen to me? So waiting for that, but if you're able to join us on the 30th, so that's our Halloween celebration, we're going to talk about just general aspects of spider silks and what we learned from that paper and what we can take that into our own research. That was, yeah. Any questions?
Any reason why spiders versus silkworms?
Yeah, so their silk is a lot more strong and elastic, and because it has, so it's made up of like 4 common repeats of amino acids, which is why it's really hard to synthesize. We learned that the hard way with Twist when they told us we couldn't do it. Which, if you actually rearrange them, it'll make it stronger or it'll make it more elastic, so there's an engineering component. But in general, it is a lot stronger, and mostly a lot stronger than silkworm silks. Yeah.
Is the molecular pathway for spider silk generation well defined, or is it a black box?
It's pretty well defined, because I would say there's a lot of research in orb weaving spiders, particularly like 15 species of orb weaving spiders. So when we think of spiders in general, we think of like the web, but only 20% of spiders make webs. So there's a lot of spiders out there that have silk, but they don't make webs, but they're used for really intricate and curious like ways. But because all spiders of that order make silk, it's pretty well documented, yeah. But that's the hard part, because their spinnerets, their glands are sensory, so they can actually change pH if they need to. So imagine like making this material machine that can sense the pH of whatever the protein is within it, change that pH, and then apply the pressure in order to make it its own material. So, I mean, they've been around for hundreds of millions of years, so obviously they've had time to perfect it, but we just haven't gotten there yet.
So how fast does this silk degrade, like due to normal?
I'm not sure. That's a good question. Yeah, when it's within the body.
Or like outside, I don't know. Like, I see spider webs, but how long does it last?
Yeah, I'm not sure. That's a good question. In the presence of water, it does shrink, and I know that makes degradation happen faster. And that's been a big issue with it being a textile, is because when you wash it, you can imagine your shirt's like this big, which isn't ideal, but I'm not sure how long it degrades on average, yeah.
Yeah, I'm looking for a liquid version that I can carry around on my wrist.
I'll let you know. Email me. Yeah.
What's your favorite spider?
My favorite spider is the wolf spider. So wolf spiders have silk, but they're not orb weaving spiders. If you want to catch wolf spiders, go in your backyard at night and shine a light. You'll see sparkles. That's actually their eyes reflecting the light. They have really great eyesight. They're called hunting spiders, so they like burrow at night and then come out and then will hunt. And then they use their dragline silk behind them to tell them how to get back home. But they're super cute. I wish I put we have one here. I wish I put a picture of one. But if you go to eml.pubpub.org, you'll see it. But they're not the sweetest. They're a little skittish, but they have good hearts.
Oh, they're cool.
Thank you, Jen. Thank you, Jen. If you're interested in joining any of the community projects, I have posted the information which you can contact them. I'm going to leave it up for a couple seconds if you want to snap a quick picture. Up next, I think Layla is coming up. Layla?
Layla.
All right, Layla.
Layla, our education manager.
Wow. Hey everyone, I'm Layla. I'm the education manager here at Genspace, and what that means is basically I handle all of our adult ed classes and our youth programs. So before I get into classes, I'm going to quickly do a plug-in for BioRocket, which is our summer research internship for high schoolers. So if you know a high schooler, have a high schooler in your life, the application will open up in November sometime, and it's basically a 6-month experience where they get to do biotech with us here in the lab. So last year we did molecular cloning, we did some CRISPR with the kiddos, we took them out to the field, so they get a really nice, well-rounded experience in the lab and also out in the field. So if you know any of them, keep an eye out for our application opening in November. Date to be determined. And now going on to our classes. As I mentioned, I manage all of the adult classes that we offer here at Genspace. So if you're interested in taking a class, for example, we have 3 main buckets of classes that we have here, and it's basically any lab intensive. So let's say you want to get to know the lab, you want to know a little bit about molecular cloning, you want to know of biotech, you know, what kind of techniques to use in the biotech world. We have classes that offer different classes depending on the content. So we have, for example, our Biohacker Bootcamp, which is like a really introductory class to molecular cloning and also a little bit of genetic ancestry. Then we have our CRISPR class, which is then going a little deeper into like how CRISPR works and really, you know, what uses and where, you know, science is taking this technology. And we also have our synthetic biology class, which is also another way of seeing how we can use biotech for different things. The synthetic bio class really focuses on using yeast, and when you change something in the genome, how it can change to different colors. So we have those 3 main lab-intensive classes that we're actually offering in the next few months. So if you want to sit into any of those classes, feel free to sign up. Another bucket of our classes is our bioart classes, where we do things like using mycelium to sculpt, right? And really the applications of the science and the art coming together in a really beautiful way. So we have those, and then we also have our bioinformatics classes, which is really using some open source, you know, data out there and using things like Python, R, or any other, you know, open source to look at sequences, or using DNA and really seeing how you can use them to analyze different sets of data, whether that's in ecology applications, medical applications, and any other, you know, ways that you can use bioinformatics. So those are the 3 main buckets of classes, and soon, hopefully, we'll be offering some more ecology and evolution classes, so stay tuned. We love doing mushroom walks, we love going out to the field. My background is in ecology and evolution, so anytime I get out in the field, I'm out there, so stay tuned for that. And yeah, if you have any questions on any classes, or if you're interested in potentially teaching a class, we are welcome to, you know, any new instructor, and if you have a specific topic, content area, we're always looking to bring in more fresh content to our community. We really want to make science accessible and really, like, you know, have people be able to take a short class to get the introduction into molecular cloning, or really, you know, open the doors for people to step into this world of biotech, or using science for art, right? And really combining all of these different aspects to make either sustainable materials, or just to know more about the world of synthetic bio, right? So if you have any questions, I'll be here. You can ask me them. And yeah, I think that's it.
And last but not least, we'll have some closing remarks from our executive director and the development manager.
Hello, online. Hello, everyone. I'm Vanessa. Huh? I always forget about this.
So first of all, we want to say thank you everybody for coming. This is a packed house, and since I've been at Genspace, this is the most people I've ever seen. And let's take a open laugh. So thank you so much for coming. This evening is meant for us to mingle, catch up with friends that we haven't seen it for a while. We're gonna do some lab tours and chatting with our presenters and staff. And we'll be closing starting around 9 p.m. So if anyone's available to help us clean up, we're like 6 people, so please help. Please stick around. Many hands make a light work. And as always, we're forever grateful for you being here. But first, we have to give a few shout outs and thank yous before we end the evening, because we are a community and we have a lot of help in making all these magical things happen.
First off, thank you to Giovanni for putting this event together. And thank you to our intern Anas and our volunteers Adil, Epi, Alex, Steven, David, and Jazz for making this night very special. Thank you. Thank you also to Aya and to Sarai for their help with the live stream and the photography. Thank you.
And finally, we could not do this work without the generous support of our donors. So thank you especially to those who have donated tonight to provide all these delicious snacks and treats and drinks, and to our government, university foundation, and corporate partners. And our profound gratitude goes to the teams at the Siegel Family Foundation Endowment and the Pinkerton Foundation, Con Edison, Brooklyn Community Foundation, the National Science Foundation, and Iveric Bio, and for the iGEM team. Special thanks to Lada Emma Leonova and Lazard Asset Management for helping with the iGEM team.
Yeah.
Thank you. So now Beth and I are going to do a special unveiling. This is so exciting. So, ready?
Yes. Good.
Ready, James?
Yeah, go ahead, James.
1, 2,
Drum roll.
3. We have a
We are super thrilled to announce that Genspace has been awarded a grant from the Illumina Corporate Foundation for our youth STEM initiatives and for other research that's happening in the lab. With this investment in the core technology, Genspace is going to be able to elevate our learners to enter the genomics era. We're going to be able to ask and answer questions that mean something to our communities. So the award, of course, is in the shape of this beautiful, amazing new baby, our mini-seq DNA sequencer, but we also get base-based DNA sequencing analysis credits and service contracts and sequencing reagents in the lab, so we can actually make this stuff happen for real. Board member Janina Jeff for helping make this happen. We're really grateful for you. Thank you, Janina.
Thank you, everyone. So now it's time to mingle.
Have fun. Good night.
And keep in touch. Please follow us on all social media platforms and Facebook.
Yeah. Please feel free to hang out.
All right. Thanks, everyone.